So we have a question from the virtual attendee. They ask, can all of the presenters please give a short timeline on the next steps? Respectively, all going well, when do you expect to have a commercial product available for treatment?
I think, as I mentioned, for Pombiliti and Opfolda, we have an approval for some adults 18 and older, and we're working on some pediatric studies. We don't have exact timelines, but those studies are ongoing and recruiting.
I think for us, we're so early on, it would be not good to try and give a timeline for that. And after yesterday, Mark told me he wants no other ERT therapies being clinically tried right now. No, so.
But it wouldn't stop us, right?
No, not stop us. So really, if I can, this is an ERT that I showed, but we also think this applies to the gene therapy and the amazing work that's shown there. We think it's complementary to some of the work that was shown here. But we really want to build the foundation for that. Our goal would be to be in the clinic next week, but we have to have the money, the resources, and the data to show that. So it's probably many years away, or 3-4 years.
So for the gene insertion for GAA, we're hoping to have that into clinical trials in the next 1 to 2 years.
For AT845, the gene therapy, AAV gene therapy, as I mentioned, we're still in phase 1-2, and that will have to be followed by a larger phase 3 study. So I would say a few years. That's the closest I can get right now for that.
And for the lentiviral gene therapy, we are now preparing for clinical trial, and we hope to start the in-human testing as soon as we can. But this will be in the coming few years, I think, too. And then hopefully we can move fast to the next phase and to market approval.
I was a history major in a lot of.
Yeah, look.
Wait for the microphone, please.
Is that on? So is the takeaway is you don't have to do ERT anymore, and you stay stable, basically? Is that the message that we're getting?
Yeah, based on the data, obviously it's early. It's only 4 patients that I showed data on. What we're seeing is, in general, stability of those the different things that we looked at, the pulmonary, all of those things. That's what we're seeing right now. We don't know what will happen in the future, or the durability. That kind of thing is always something that you think about in gene therapy. Thank you.
I have a question here.
Oh, can I just follow up on that real quick? The hope, gene therapy, lifetime developing, but even if it starts to wane over time, as we learn more and stuff, there's nothing that would prevent an ERT to go back into it, like to raise the levels. So I don't think gene therapy and ERT has to be exclusive. The goal is for it just to be the gene therapy approach, but if we needed to, we could come back up with an ERT to make sure we're getting enough.
Yeah, I can add to that. There will be some variability among patients, so then it will be very good to be able to treat also with enzyme therapy, maybe at a lower dose and a lower frequency, but this is something to keep in mind.
Thank you. Here you go.
Hi, Ryan Colburn. As an education request for all of us, can you clarify or differentiate the role of like I guess, let's say the recipe for the GAA protein used in each treatment and versus the elements that are specific to the delivery, so vectors, tags, promoters, insertion scripts, et cetera. And how should folks or patients or physicians in the room rate this? And is that understanding useful to evaluate different options that are on the market or in development?
Well, that's a typical Ryan question. So, Part one. No, speaking for myself, for me, I kind of consider the protein backbone for what I describe, and maybe some of this, to be very similar, unless we're putting a tag on either side of the GA to help target it in the gene therapy. What I'm looking at is the carbohydrates that are on there and stuff for an ERT, and that's what really the other generations are also trying to do. Now for the gene therapy, there's a lot of difference in probably secret sauce about promoters and how they make, so that's I don't know, I don't want to speak for people, but that's kind of part of the magic. But for me, I kind of consider the protein, the carbohydrates is what I'm trying to affect. Or, like the IGF-2 approach uses the same receptor. It's the other name for it's the IGF-2/CIMPR receptor. So that's kind of nice. It's all kind of in that same world. It's just a different portion of the receptor it binds to. So I kind of look at protein that acts to break down glycogen, and then I look at the carbohydrates or the other targeting motifs to get it to where it needs to be. So
Maybe I could just add to what Russell said. He showed really nicely in his talk about the first enzyme, myozyme, and that only a quarter of it actually has a moiety on there that can mediate the uptake. So I think we know that if you, and we've seen now coming out from clinical trials, that if you can increase the amount of that protein that can get targeted, that will really help. But I think it's a little too early probably to say really which approach is going to be the best, but it's something that you do need to have an approach.
Maybe to clarify, because I didn't have time to say it. So there's a receptor called mannose-6-phosphate receptor, which is the other name is IGF-2 receptor. And there's a binding domain for mannose-6-phosphate, and that's that's one strategy to have the proper mannose-6-phosphate residues on the enzyme to be able to bind to that receptor in a proper way. That's one mechanism. The mechanism that we are using is we are not relying on mannose-6-phosphate, we are relying on another part of the receptor that can bind IGF-2. And the affinity for IGF-2 is higher than for mannose-6-phosphate. That means that we bypass the mannose-6-phosphate, we don't care anymore, and we use the other part of the receptor, and by that we improve the uptake by cells. So that's a different concept.
Katherine, maybe you will tell about CD63 a little bit, that it's another way of delivering the...
Yeah, so CD63 is a tetraspanin protein. It's not classically a receptor, but it came to my interest because it traffics other proteins to the lysosome and actually controls the degradation of a couple of other different kinds of proteins. So we got interested in this protein as a potential alternate way to get into the lysosome. And in particular, we were interested in it because it has a broad expression pattern, but it's not highly expressed in the liver.
And a quick follow-on for Dr. Pijnappel. The biopsy slides that you showed and demonstrated correction on look to be from a very early state of disease progression. Do you have any experience with a more advanced state of tissue damage?
These are mice biopsies, and actually this is what you see if you go look in the mouse muscle. So it was not really early, it was 8 months. So this is late adult for a mouse.
I guess early from the standpoint of the white dots were small and well distributed as opposed to what we see in more advanced human progression.
Yeah, I agree. So the phenotype in human is more severe than in mice. The reason for that is unclear. Mice are stronger, and you see that also for other diseases. Yeah.
We got the mic over here.
Now, my point was, and I have a question, I'm over here, Hans van de Ploeg, and I had a follow-up questions to the one question asked in the back, whether you can stop ERT. What I saw in your presentation is that 3 of the 4 patients stopped ERT in your trial, and I wondered, maybe I missed it, did the patients stop ERT once they received the AAV gene therapy, or did they continue for a while, and did you then decide to stop? And also the second question, maybe I saw you had 2 different dose groups of viral load. Do you have an idea already how to proceed?
To answer the first question, so they were stable on ERT. It was up to the physician and the patient when they stopped, but they did continue for a period of time after they were dosed with the gene therapy, and then they discontinued. One patient has not discontinued, and that's the decision that was made with her physician and herself.
So the data shown are all without ERT for 3 of 4
3 of the 4, and there is 2 different doses. So we haven't, again, it's early, but there doesn't seem to be a huge difference that we're seeing at this moment, but that might change in the future.
But we need to make clear comment for the whole group that gene therapy is not working exactly on the day you administer it. It's not like a ERT. So there's a phase of at least 4 weeks you need an overlap.
I have a question over here.
So first of all, congratulations for excellent work. I had a question for Katherine. What is your AAV serotype that you're considering, and would you be excluding some patients because of pre-existing antibodies? And then I have a question for Shannon about the conditioning or the regimen that is being used to prevent the immune response. And you did show data on 6-minute walk test and FVC, but I don't know if I missed it. I didn't see any biomarker data to see how it's going. And a question for M6P, for Russell. You're showing the comparison to Myozyme. I think it would be very important to now compare it to the next generation therapies because we are in that phase already right now. Thank you.
The serotype that we're using is AAV8. We will most likely have an exclusion criteria for people with pre-existing anti-AAV8 antibodies. I do want to say I think the field, the gene therapy field as a whole, is not, we're not happy, we're not satisfied with excluding patients from trials, and we're not satisfied with excluding patients from therapy. And we're working very hard on other immunomodulation regimes that would allow patients to get therapy even if they have pre-existing antibodies. And we're actually, next week, a lot of us are going to ASGCT in Baltimore, and we're going to be talking about a lot of that. So just wanted to add that.
Thank you for the question. Shiri, I wonder if you could answer. So Shiri is if she could get the microphone back there. She's the clinical lead for the Pompe program, so she would have a better idea about the biomarker question.
Thank you for the question, Priya. I'm a medical lead for this program, so I would like to take the question. So the first question was about the, biomarker? So the patient started taking a corticosteroid 1 day prior to the ADF5 dosing, and after that, patient keep taking the highest dose for a while, and after 8 weeks of the high dose, and after this, and then started tapering up the steroid. So, and then as Shannon showed it slide, most consistently observed adverse event was AST and ALT elevation. No, we haven't seen GGT elevation, so only seen AST and ALT elevation. But all the events were very responsive to this threat, so there's no additional immune responsive agent.
What I meant was the CK and the immune score. You showed 6-minute walk test.
Yeah, so biomarker was shown. So even after the ERT cessation, both CK and HEX4 has been on the stable dose. So we haven't seen any elevation spike after the ERT cessation in the other patient who discontinued ERT. Yes.
So, I wanted to say I wanted to give a really nice shout out. We're the industry. We're not, we get along. So I wanted to also say, you asked about the drugs. It's really neat. Both Amicus and Sanofi have made their drugs available for us to start doing the comparative studies in these to really line them up, not like the clinical trials that are all done differently. We'll do them in the same study, the same animals, dosed the same way with the same endpoint. So we're really excited about that. And I think it just speaks to, I think the industry goes, we do need to figure some of this out, and they're willing to work together to do that. So wanted to make sure. Yeah.
Yes.
By world? Oh.
Over there.
Okay, so I just have a question. There seems to be a lot of receptor. Okay, you gotta be. Right. I'm just wondering, are you?
No, we can't hear.
We can repeat it if you want.
Okay, is it working? Okay, so there, that transfer.
We can hear it. Yeah, go ahead. We can hear it.
Okay. Are you seeing increased transcytosis through muscle capillaries with the transferrin receptor? Are you seeing increased GAA activity in muscle using your approach?
So the question was, with the transferrin receptor targeting, do you also see increased transcytosis in the capillaries into muscle and correction of glycogen storage in muscle? And the answer is yes, we do. I think the transferrin receptor is working in two ways. As far as the antibody, it's hitching the transferrin receptor, which brings iron from the bloodstream into tissues across the blood-brain barrier and also across other capillary barriers. So in our experiments, we do see much better glycogen reduction in tissues with the transferrin receptor targeting, and I think it's a combination of, or rather we think it's a combination of both getting out of the bloodstream, transcytosing out of those capillaries, and then also uptake into tissues once it gets across the bloodstream and is in contact with cells.
Have you looked into other transcytosis receptors that might be more prevalent on muscle capillaries?
We have looked at, in terms of muscle uptake, not muscle transcytosis. We've looked at a handful of different receptors, and we haven't done like a real rank order of like how much transcytosis there is.
We have a question over here.
Hello, my name is Scott Eggers. This question's for Astellas. For your clinical trial, how are you modulating the uptake into the liver, avoiding rejection?
We're using AAV8, and with any AAV, the liver will take up a good deal of the viral vectors. So other than doing the corticosteroids that Shuri mentioned to sort of protect the liver a little bit, we're not modulating the uptake by the liver.
Okay, and also for those that are off of ERT, what does the history look like of the continued GA activity off of ERT? And I qualify this, I'm actually a gene therapy participant myself, so I kind of look at a comparison.
Off ERT, what was the question? For those
what does their GA activity look like naturally occurring after coming off of ERT?
I'm going to give that again to Shuri, who has that information.
So our technology is to produce GAA in muscle directly. So then we are also collecting the plasma, both the plasma GAA and muscle GAA after ADA45 administration, and we have confirmed that the GAA produced in muscle cell does not have much ability to excrete into plasma. So we haven't seen elevation of GAA in plasma after treatment, but we confirmed that the GAA elevation in muscle biopsy after the treatment and even after the ERT cessation. So, and of course data, we haven't, this time we didn't show the biomarker data, and data of course looks variable among the patients, but we have seen some elevation in GA activity and production in muscle biopsy samples.
Thank you. Okay. I have another question over here. Oh, well, I have her over here first. I'll get to you.
Theme. This is Azam Gokarappan from LDRTC. So I was going to ask the area under the curve. I understand it is not excreted too much into the peripheral, but what is your muscle, the percentage increase in 12-week versus, I think, the 48-week, is that right? Any subjects reached the other goal? Could you give us an idea?
Marsha, can you get her the mic over there? Yes, please.
Thank you.
Thank you.
So one patient at the higher dose, which is 3 times 10 to the 15, the 13th VZ per gig, patient shows the elevation of about 30% of the healthy volunteer level of GAA at 1 year time point. We have observed, but in one patient, and the other patient showed numerically higher than the baseline level, but the level wasn't that high.
Okay, all right. And the other thing is about the AST, ALT elevation. And I probably may have missed how many patients developed?
Three or four.
Okay, and where is the time period? And how do you differentiate between its liver versus muscle? It may come from the muscle too. I mean, I understand it's ALT is for more liver, but there may be muscle inflammation in addition to what's happening to liver too.
So all the patients didn't have AST and ALT elevation at baseline. So none of the patients have shown the sign of liver dysfunction at baseline. And the level of increment of AST ALT is about 3 times higher the upper limit level. Because the transaminitis is the cluster effect, so we think that those are the liver.
But 3 times is pretty significant. I mean, we do multiple gene therapy protocols, and most protocols actually rely on 50% increase, and we just watch like a hawk, even though that's ALT and AST low, it's just a little bit elevation actually becomes an AZ, okay? So how do you, for these 3 individuals, so what is in relationship to the muscle enzyme GAA levels? So I'm asking about the transduction and the success of it. So basically you're going to those other cohorts, and you want to understand what's the efficacy in the first cohort to move to the second cohort, is that right?
Yes, and the level went back to the normal level within a very short period of time after seeing the very high elevation. So we haven't seen the 3 times or 2 times higher than the upper normal level of the AST ALT elevation for the 2 or 3 times point consequently. And we haven't seen the elevation of AST ALT as well as CK elevation. So the elevation of the liver enzyme and muscle enzymes are the differentiated.
I'm very sorry, but we have to move on. I think you can continue. We have a couple of other questions from the audience.
Yeah, we'll take 2 more, and then we need to cut to break.
Yes.
Thank you. Is it related to this? Okay, Okay, he was first.
Thank you. It's a quick comment and question for all the pharma. I'm fascinated as a clinician by the huge number of new avenues for the treatment of Pompe disease, but I am surprised that there is the approach of small molecules or oligonucleotides does not seem to be on the pipelines. Knowing that today, for several of neuromuscular disorders, oligonucleotides is part of some new treatment, for example, SMA. So have you any explanation or reason not targeting, for example, IVS1 mutation with oligonucleotides? Is it because of limits of a problem of production? I'm not a scientist.
I can comment on that. So we have developed antisense oligonucleotides, as you know, for the IVS1 mutation, and they work fantastically in the lab. We can completely restore Pompe disease in the lab. The problem is the delivery. So SMA has a delivery intrathecal, and that works with a certain chemistry for unknown reasons. And now they are progressing indeed for ALS, etc. So it's really promising. So to the neuromuscular junction and the brain, you can use this to the liver. Now we have the also targeting modules. This is the low-hanging fruit. Muscle is very difficult. So Pompe disease is a muscle disorder. It is very difficult to get the AONs in a muscle, and that.
Go on, Brian.
Studies with good expression in the muscle. Using transferrin receptor for myotonic dystrophy, and they really show promising results. So that's not longer true. So therefore, there's a lot of development for this, and now it would be the fusion of an antisense oligo with the transferrin receptor, and then you can tackle the splice site mutation.
Yeah, that's
a very straightforward approach.
So let's see. So the big issue there is the safety. So let's see how safe that is compared to the dose that you need for efficacy. The kidney and the liver are at risk, and because this is a continuous treatment, lifelong, you cannot stop it. So let's see how safe this it will be. But indeed, we need targeting modules, and we are also working in the lab on that. But there's nothing yet in the clinic.
We have our last question over here.
I had a question for you, Pim. With the IGF-2, there have been clinical trials with it, and we do know that there is a real risk for hypoglycemia and a potential risk also for tumor formation. Has that been factored in your molecule?
Yeah, so indeed, there has been a clinical trial for the IGF-2 tagged enzyme applied as enzyme therapy. And there have been events of hypoglycemia in the high dose, 20 mg per kilo, that has been published with Barry Byrne. And we have checked that. So you can imagine that you have a very high dose at a very short period when you apply the therapy, and then it is cleared from the liver very fast. That's a completely different pharmacokinetics compared to gene therapy. The expression in the blood after lentiviral gene therapy is extremely low, but constant, and it goes very efficient in the tissues. So that is at a dose that is so much lower that it is it will not cause hypoglycemia. Of course, that needs to be seen in humans, so we have to see. But for mouse studies, this is what we can infer. And I hope this will remain like that. If there will be still a problem, we also have some versions that can reduce the binding to the insulin receptor. So that would be the alternative.
And also the tumor risk, right? Because it's a growth stimulating factor.
And hypoglycemia was controlled in clinical studies. Yeah, it was there. But
it was
in several patients. It was controlled.
It was quite severe, and it needed treatment for it, and they had to wear continuous glucose monitors. So it was not trivial at all.
Yeah, well, let's see. And about the other safety effects, yes, we have to monitor this. And I think the expression is so much lower that I hope this will be fine, but let's see. Yeah.
Thank you. That's the last question.
Okay. Thank you very much.
Before we break, for the break, a couple of admin things. If you did not receive your welcome bag like this, please see Marcia back there and get one, please.